Europe has become home to an increasing number of new species as rising temperatures and climate change alter environments around the world. Some of these species are well accepted, others are a risk to human health and food safety or a threat to other established species. In the EU-funded Project REACT, researchers are working together to focus on 2 insect species that are likely to become invasive in Europe in the near future. The 2 species, Bactrocera zonata, the peach fruit fly, and Bactrocera dorsalis, the oriental fruit fly, could soon not only damage individual agricultural production sites, but also affect the citrus fruit trade. The goal of REACT is to find novel and, above all, pesticide-free methods to control the dispersion of both pests. In this video, we want to explain what steps need to be taken to develop such a method in a cost-saving and efficient fashion. Before we look at these steps, however, let's hear Marc Schetelig, Professor for Insect Biotechnology and Project Coordinator, who emphasizes the urgency for the approach taken in REACT and describes the way that targeted fruit fly species continue to invade European countries.
Whenever an invasion occurs, you have to find it quickly, because otherwise you cannot act appropriately. So you need a rapid detection of your pest as fast as possible, and then you need local technologies to act quickly, and of course also public awareness and programs that make it possible to use your technologies quickly to eradicate them. And the REACT project develops exactly that, from the rapid detection to technologies that can be used immediately, having a plan, and then really use that on the 2 Bactrocera species, still the fruit flies we're working on. The common ways are normally by travel, human travel, and trade, from fruit shipments, any flights, any people bringing things into the country that sometimes they shouldn't, but also by natural dispersion into the countries. And the countries that are invaded constantly are European countries in the south. Lately we even had incursions in France and in Spain, and this year, 2023, in Belgium also. So very, very northern for this hot-loving species, and we are really at risk of invasions.
To fight the threat of fruit flies invading and damaging crops, researchers from 12 countries and 15 institutions have united to perform the following tasks: environmental analysis and alignment of gene sequences, strategies to early detect invasive fruit flies, sterile insect technique, in short SIT for population management, mass rearing and treatment of flies, release mechanisms and dispersal of sterilized male flies. Let's have a look at each of these steps now. In the initial phase, we will analyze the environment and how environmental conditions match the genes of our target pests. The aim is to gain a better understanding of the conditions under which our target pests spread and how our species behave in different environments. For example, if Bactrocera zonata and Bactrocera dorsalis are able to settle in certain ecological environments, it's important to understand how they feed and reproduce on unfamiliar fruits, how they compete with other species in a new habitat, and how they adapt to different temperatures. Hélène Delatte, a researcher based in La Réunion, explains that the temperatures could continue to make it difficult for Bactrocera zonata and Bactrocera dorsalis to travel to or overwinter in Europe.
Temperature is very important, and it's a big issue for these 2 fruit flies, which are indigenous of Asia and tropical Asia. When they'll be facing Europe, they will be, at their tropical, they'll be facing a very whole range of temperature, which will be in the different seasons in Europe. So they'll be facing very cold temperatures and higher temperatures. So they might be less competitive in some part of the season, or very competitive during summer, for example, facing other indigenous species.
To understand the nature of fly species, it is then important to study their genes and add to existing genetic databases. In addition to careful analysis of gene sequences, this task also involves the specific monitoring of certain external conditions, such as the larva's diet. Francesca Scolari looks into the chemical processes that affect the species' tissue, organs and cells.
So the aim of my work is to develop a novel approach to evaluate the quality of the flies that we produce for the sterile insect technique. The study of the chemicals that are present in a specific tissue can help because they can be identified, quantified, compared with other treatments. So the idea is to then use this knowledge to create, for example, supplement for the diet to make these flies more fit, more competitive, and work better in the field.
Now that we have a basic understanding of the composition and behavior of the fly species that REACT is designed to control, we are turning our attention to practical applications in the second phase, detecting the presence of invasive fruit flies. The aim of this phase is to develop tools that help identify the key invasion facilitators and drivers at the early stages of invasion. While the overall process is similar to that used in the well-known COVID-19 tests, Gurpainz explains how CRISPR-based diagnostics will be used in REACT.
CRISPR is very well known for the genome editing properties, but in this project we don't use CRISPR for genome editing. We use it just exclusively for detection. And the way we do that is when we find larvae and we don't know exactly which fruit fly this is, because at early stages they all look very similar or almost identical. We squish them, and then we apply the CRISPR mechanism that eventually, if it detects a specific sequence that we know that belongs to a specific pest, then we get a fluorescent light. So this is how the detection works.
Once researchers like Gur have identified the flies, the next task is to treat sterile flies to prevent the pest from spreading in infested areas. This involves several steps. First, the female pupae is separated from the male pupae in the laboratory. This is done by machine and based on the color of the pupae. Male pupae are brownish dark, while female ones are white. Next, the male pupae are exposed to radiation and sterilized. When they mature, the sterilized flies will be able to still mate but cannot produce offspring. In a third step, the sterilized male flies are released in a pest-infested area. Despite mating, the flies only produce eggs that contain undefined cytoplasm. So while the mating process and subsequent egg laying itself will still take place, no new larvae will develop from the eggs. This helps in gradually reducing the pest infestation and stopping its spread. Creating the necessary SIT procedures, the next step is to improve the breeding facilities for the pests targeted by REACT. The mass rearing of sterile insects is by no means new. The worldwide directory of SIT facilities, maintained by the International Atomic Energy Agency in Vienna, contains 75 entries for facilities that breed insects for autocidal pest control. For successful rearing of Bactrocera dorsalis and Bactrocera zonata, it is important to establish the ideal temperatures for pupae and flies, construct effective cages at a low cost, and implement appropriate feeding techniques. By analyzing the data from the early stages of the process, it is possible to create a rearing environment that will ensure an adequate number of flies for release. The fifth and final step is the release of male-only sterile fruit flies. As well as the logistical issues of releasing flies on the relatively small plots of land typical for European agriculture, there's also the question of the actual biological quality or fitness of mass-reared and sterilized fruit flies. Antonio's Avgoustinos outlines their ideas for assessing the quality of fruit flies and for release mechanisms using another already established fly in the region as a proxy for the REACT target pests.
Our main question is what the sterile males that we are going to release, what they will actually do in the field, how they will perform. So in the first set of laboratory experiments, we can check their flight ability in specifically designed flight meals, their survival under stress conditions, and also their survival or longevity under no stress conditions when we provide them food and water. Later on, when we want to go really in the field, we have to do larger scale experiments, open field or semi-field experiments. There we test actual parameters in the field like dispersal of the flies, We need to monitor how much they can fly and how much they will survive in the field. And also in semi-field experiments, we can see if they are competitive to wild males. So we see how much they mate and if they manage to suppress the natural populations under contained conditions.
As we have seen, REACT is developing a step-by-step plan to eradicate insect pests and help farmers protect their food production and livelihoods. REACT researchers analyze and align gene sequences, develop tools to detect invasive fruit flies, improve the use of SIT for population management, manage the mass rearing and treatment of flies, and test different release mechanisms. These efforts may not only help prevent the spread of the Bactrocera species, but also help control other troublesome pests in the future, leading to a greener, pesticide-free environment.